Formed elements are the cells and cell fragments that are produced in bone marrow that circulate throughout the bloodstream. The first category under the heading of formed elements are the red blood cells, or erythrocytes. These are by far the most abundant formed elements, and they function to transport oxygen throughout the body, but also help transport carbon dioxide throughout the body. The next category include the white blood cells, or the leukocytes. And leukocytes are often categorized as either granular leukocytes or agranular leukocytes. Granular refers to the granules or the vesicles that you can see inside the cells when stained and viewed under the microscope. So these drawings of the white blood cells are after a staining procedure, which is often the Wright stain. The Wright stain uses a couple different dyes to be able to differentiate the different leukocytes. But one way you can tell that there's a difference is that some have these little vesicles inside of them that you can see, and some do not. Let's look at the granular leukocytes. They include neutrophils, eosinophils, and basophils. We're going to be looking closely at each one of these in a moment. The agranular leukocytes include the lymphocytes, which there's a number of different types of lymphocytes, including T lymphocytes or T cells, B lymphocytes or B cells, and natural killer cells. Monocytes are also an agranular leukocyte. You'll notice that each of these have a different color to them. They have different looking nuclei, so these darker stained structures within are the nuclei. They may or may not have granules, and those granules may or may not be stained differently. Finally, the last category under the heading of formed elements are actually not cells, they're fragments of cells. Platelets are derived from big giant cells called megakaryocytes, but then they fragment into little portions of the cell, and these are used in your blood clotting mechanism. So this is just a heads up on where we're going next. We're going to cover the red blood cells, the different types of leukocytes, and then finally platelets. Before we get to each of those, we're going to just highlight this very complicated figure. This figure shows us the formation of the different formed elements. So you have to imagine you are in red bone marrow. Red bone marrow is the site of hemopoiesis. Hemopoiesis is the development of formed elements in red bone marrow. Hemo means blood. So your blood is formed inside the bone marrow. These cells and cell fragments all originate from a certain type of cell, stem cell, called a pluripotent stem cell. A pluripotent stem cell has the capacity to develop into many different cell types. In fact, you'll see from this figure that a pluripotent stem cell could eventually become a red blood cell, could eventually become a cell known as a megakaryocyte that fragments into platelets, could become an eosinophil, a basophil, a neutrophil, a monocyte, lymphocytes, and natural killer cells. So these are very useful cells, in that they have the ability to develop into any of our formed elements. What drives this is often hormones and other chemicals. So the environment in which these cells are in contain different chemicals that develop these cells into specific formed elements. One other important step is when a pluripotent stem cell differentiates into either the myeloid stem cell or the lymphoid stem cell. Once a pluripotent stem cell differentiates into a myeloid stem cell, we know it's either going to become a red blood cell, platelets, or these four white blood cells: the eosinophils, the basophils, the neutrophils, or a monocyte. If it differentiated into the lymphoid stem cell, it's going down a different path of development and will become either a lymphocyte or natural killer cell. So this process is called differentiation, in that an undifferentiated cell, meaning it really hasn't had a function yet, becomes specific cells dedicated to specific functions. Again, this is all happening in your bone marrow. These cells are going to be released from the bone marrow. Some of them will end up going to certain tissues throughout the body where they will function. This is all promoted by hormones and cytokines. Cytokines are chemicals that help stimulate cells to become certain cells. Let's begin by looking at erythrocytes. Erythrocytes are red blood cells. They are the most abundant formed element. They have 5 million per microliter on average. Males tend to have slightly higher numbers of red blood cells than females, but on average we can use 5 million as an average. And that's per microliter. Microliter is a very, very, very small volume. They have a certain characteristic. Obviously they are red because they have a pigment that reflects the red wavelength. They're about 7 to 8 microns or micrometers in diameter. That's fairly small in the world of cells. They have a certain shape, biconcave discs shape. They lack nuclei and many other organelles, and they have a finite lifespan, meaning they're not going to divide into more and more red blood cells. They're going to circulate in the blood for about 100 to 120 days, and then they need to be removed from the bloodstream. So let's look at the shape of a red blood cell. This biconcave shape has to do with the fact that through its development, it loses a nucleus that was taking up space. It loses a lot of the organelles and flattens. Why is this shape so important? 2 reasons. One, it's very small and flexible and can fit through very, very small capillaries in the body. 2, it has a very high surface to volume ratio. That allows oxygen and carbon dioxide to enter and exit a red blood cell very efficiently. So red blood cells cannot be spherical because oxygen would not be able to come in and then exit very quickly as they run through capillaries in the body. So the shape of a red blood cell is extremely important for its function. What is its function? Well, it contains hemoglobin. And if you know what hemoglobin does, you'll know what the function of a red blood cell is. Hemoglobin is a fairly complicated protein that's used to transport most of the oxygen and part of the carbon dioxide in the bloodstream. Let's look at the structure of a hemoglobin molecule. Shown here in this drawing, you can see a blue kind of ribbon-like portion of the hemoglobin molecule. The blue portion of the hemoglobin molecule is known as globin. Globin is the protein portion of hemoglobin. And in fact, there are 4 globin subunits. There's typically 2 beta globins and 2 alpha globins. So this hemoglobin molecule, you should be able to notice it has 4 units to it that look very similarly, and then those 4 units come together to form a single hemoglobin molecule. So a hemoglobin molecule has 4 globins. Each globin contains a heme molecule. Heme is what we call the non-protein pigment. It has kind of a ring-shaped structure to it. At the center of heme, you can find an ion, specifically iron, or Fe. Iron is the atom that will bind a single oxygen molecule. So if you look at the entire tetramer of hemoglobin, tetramer is a 4-unit molecule, you can see 4 globins, 4 hemes, 4 irons. Therefore, a single hemoglobin molecule can transport 4 oxygen molecules. This slide looks extremely complicated, but don't be too scared. We're going to walk through it very slowly so you can better understand the life cycle of a red blood cell. This figure starts over here at removal of red blood cells. I would prefer to start at number 8, because number 8 shows a bone, and you might recognize this bone. This is the proximal end of the femur. This is the head of the femur. The reason they show that is there's a lot of bone marrow in that proximal end of the femur. And inside that bone marrow, there's a process called erythropoiesis, which is the formation of red blood cells. Notice some of the things you need to produce a red blood cell. You need iron, you need globin, you need vitamin B12, and you need a hormone called erythropoietin, or EPO. If you have all the right ingredients, and there's more than these 4, red bone marrow can efficiently produce red blood cells. These red blood cells are going to circulate for about 120 days. They're going to get worn out, they're going to get old, and they need to be removed. So the next question is, who removes red blood cells from circulation? The answer is typically your spleen, but your liver can help out as well, and bone marrow. So individuals that have to have their spleen removed can still remove worn out red blood cells by using their liver and bone marrow. So these guys are the old red blood cells that already did their job. What are we going to do with them? Well, it really has to do with what are we going to do with all that hemoglobin. Some parts of the hemoglobin we can recycle. For instance, we can take the globin proteins, break them down into their amino acid components, and reuse those amino acids. So the body's very good at recycling protein. The next component is the heme that has the iron attached. The iron can get removed, transported using a protein called transferrin, transported to the liver and stored. The protein ferritin can store iron. Then, if we need more iron, say for example in our bone marrow for more red blood cell production, we can transport that iron back to the bone marrow and reuse the iron when we synthesize new hemoglobin protein. So iron can be recycled. The component of hemoglobin that cannot be recycled is the heme molecule. So heme must be metabolized, and then eventually the metabolites are excreted from the body. So heme undergoes a lot of changes, and we call those changes metabolites. Heme gets first metabolized into a green pigment called biliverdin. It gets metabolized into kind of a yellowish orange pigment molecule called bilirubin. bilirubin is transported to the liver, and the liver will help remove bilirubin from the body by putting it in bile. You've probably heard of bile before. And we're going to be talking more about bile in later chapters. But bile has two purposes. One, it helps in your digestion. It helps in your digestion of fat. Two is it has an excretory component. It has something in it that we need to get rid of, and that is the bilirubin. Bilirubin is eventually placed into your small intestines because that's where bile goes. So it goes within bile to your small intestines. In your small intestines, bacteria metabolize bilirubin into other metabolites that reflect other wavelengths. So for example, stercobilin is the major pigment found in feces. So the color of feces is largely due to these metabolites eventually coming from heme. Some metabolites are reabsorbed into the body and then eliminated in urine. So ultimately, this heme molecule is removed from the body either as a metabolite in urine or a metabolite in feces. So that might have been very complicated, but trust me, if we go through this process step by step, just think about what we need to do with all this hemoglobin from all these worn out red blood cells. We really have to remember that some components of hemoglobin can be reused, like the globin and the iron, but it's that heme that gets a little tricky. We can't reuse that heme. So we have to figure out a way to get rid of it. And your liver has really figured out that way by putting it in bile. The next video, we're going to continue with red blood cell discussion by talking about some of the problems that might arise if we don't have enough red blood cells or we have too many red blood cells.